Our last video gave you a tour of the carbohydrates, and I left you with a bit of a conundrum, which is, although carbohydrates are a convenient way for cells to store energy, they come at a cost because they're bulky. Our next type of polymer offers a solution to that problem, and those are the lipids. But actually, I should throw in a small disclaimer here, because lipids generally don't get big enough individually to be considered a full polymer. However, they are a major type of molecule found in cells, and they can be both incredibly diverse and incredibly important for determining cell function, so it's a good idea to understand how they work anyway. So we'll just put polymer in quotes. We're going to start out by examining the group of molecules that are known as fats. Fats are built out of two different types of building blocks. The first building block is going to look a bit like a sugar, but with something missing. So here's what that one looks like. So, do you see what it's missing compared to a sugar? This molecule is known as glycerol. And you'll notice that there are these three hydroxide groups here hanging off of this glycerol. So as with the sugars, the loose hydroxide groups are an opportunity for other things to link up to this molecule. And specifically, here's an example of another molecule that can hook up to this glycerol backbone. So you'll notice that this molecule consists of a long chain of carbons, and in this case all these carbons are bonded together with single bonds. And then on the business end of this carbon chain, so what have we got here? So if you said, well, we've got a carbonyl plus a hydroxyl group, and so then we've got a carboxyl group. So if we put these molecules next to each other, you might notice something that looks similar to some things we saw for the carbohydrates. So what can happen here is another example of a dehydration reaction to link these two pieces together. And the outcome is something that's going to look like this. Goodbye, water. And so this outcome is something that is called an ester linkage. And again, don't get too bogged down in the name. I'm not too concerned about knowing that this is an ester linkage or being able to draw this whole thing out at a cocktail party, but at this stage it's more that I want you to be able to recognize this as signs that you are looking at a fat. And so notice that there are these three places where things can hook into this glycerol molecule. And the molecule type that's hooked in this case, is a molecule that's known as a fatty acid. And so notice that in contrast to sugars, this fatty acid consists mostly of just carbons and hydrogens with this carboxyl group at one end. So that's in contrast to that CH2O ratio that we saw with sugars. So a really cool thing about fatty acids is that they can vary, and they mostly vary in two ways. First off, they can vary in length. So here I've had to abbreviate it, but this 16 carbon fatty acid that I've drawn is a special one, and so this 16 carbon fatty acid is called palmitic acid. And another really common one is an 18 carbon one that's called stearic acid. The second way that fatty acids can vary is in the presence or absence of double bonds. So neither palmitic acid nor stearic acid has any double bonds. So these are both said to be saturated fatty acids because there's a hydrogen hooked up to every carbon at every single point along this chain. And in contrast, fatty acids where there are double bonds present are generally referred to as unsaturated fatty acids because there's points where there are hydrogen atoms that are missing. Yeah, something like that happens. So now we're going to think back to something that I talked about during the lecture on carbon chemistry, and that is about the consequences of having a double bond present in a molecule. So that double bond is going to be stiff, right? And also that double bond can cause things to point in one of two different directions. So now I'm going to draw out an example of a fairly common type of unsaturated fatty acid. And it's actually called a monounsaturated fatty acid, oleic acid. Okay, so with oleic acid, what you'll notice here is that this is another 18 carbon chain. And there's a double bond present here between carbons number 9 and number 10. So I suggest that you pause for a moment here and note whether you think this is a cis bond or a trans bond. And now I'm going to spoil it. I hope you correctly identified this as a cis bond. So that makes oleic acid what's known as a cis fatty acid. One of the consequences of a double bond like this is that unsaturated fatty acids can't pack as closely together with other neighboring fatty acids. They have a kink in them here. And so a larger consequence of something containing a lot of unsaturated fatty acids is that that substance is going to actually wind up having a lower melting point. And that's why a lot of unsaturated fats, such as olive oil, wind up being a liquid at room temperature. And in contrast, a saturated fatty acid with a straight chain like this, those chains can pack together very closely. And so these tend to have a higher melting point, and so they tend to form solids at room temperature. And now that I've explained these points about fatty acids, you're in a good position to notice something. Have you ever looked at a food package that proudly declares no trans fats and wondered what that was all about? Well, it turns out that historically, people developed synthetic processes to convert unsaturated fats to saturated fats, basically by bombarding them with hydrogen to keep the fats from then separating out in liquid form. So think about something like a greasy potato chip, trying to avoid that problem. However, the processes that are used to convert unsaturated fats to saturated fats also wind up producing trans fats. And the thing about trans fats is, as I've noted already, the shape of a molecule can wind up having a huge impact on how it functions inside of a living thing. And since our cells only started to encounter trans fats after we figured out how to make them chemically by accident, our cells haven't had time to come up with effective ways to work with trans fats. So people who eat a lot of foods that contain trans fats will wind up having a buildup of trans fats throughout their bodies, and then these trans fats can start to interfere with a whole host of biological processes. So I'll leave it up to you to think about trans fats. And now, you might also have heard of another type of fatty acid called an omega-3 fatty acid. So this group of fatty acids is polyunsaturated. So there are multiple double bonds that are present, and they're distinguished by having one of those double bonds at the carbon that is 3 atoms away from the fatty acid's terminal methyl group. And so the terminal methyl group, so opposite from where this carboxyl is referred to as the omega end. So that's the Greek letter omega, aka the end. So for example, why don't I draw out linoleic acid for you? Okay, so linoleic acid is 18 carbons long, and as you note, there's 3 double bonds here, and they're cis double bonds, so this is polyunsaturated. Again, as an omega-3, there is a double bond at the 3rd carbon up from this terminal hydroxyl group here. And I'm just going to note that there's generally some reasons to think that there are benefits to including omega-3 fatty acids in your diet, but I kind of see that as a side note to the general recommendation to eat a plant-rich diet that contains lots of fresh fruits and vegetables. Okay, so just to recap, again, so fats can vary either because of variation in the length of each fatty acid or due to the presence of double bonds. And that all affects how those fats wind up behaving chemically. And I just want to make 2 other notes here about fats, and then I'm going to briefly talk about a couple of other related molecules. So with just carbons and hydrogens here, that's going to mean that these fats are going to be non-polar. And if they're non-polar, what does that mean for how they're going to behave when they're put in water? It means that they're not going to dissolve in water. So fats are considered to be hydrophobic. So like a phobia is a fear, so what this literally means is water fearing. The other point is that these hydrocarbon chains are also a way for cells to store energy, and they are much more compact than carbohydrates. And in fact, 1 gram of fat contains 9 calories of energy, while 1 gram of carbohydrate contains only 4 calories. So that's the fats in a nutshell, but there are 2 other special categories of lipids that I'd also like to mention. The first category is another type of molecule that's also built off of this glycerol backbone, but in this case, one of the 3 bond sites has a replacement molecule instead of a fatty acid. So I'm going to draw this whole thing out. Okay, so here's a replacement molecule, and up here, and just want to ask, do you recognize this? And this P in the middle surrounded by O's should be a giveaway. This is a phosphate group, and this has some cool implications. So notice the negative sign up here. So phosphate groups, I hope you remember, are polar. And so because of this, this phosphate group tends to be rotated away from the nonpolar fatty acids, as I've illustrated here. So this whole molecule winds up being a really interesting hybrid molecule because it has a polar head and then a nonpolar tail. And collectively, because it has a phosphate group, but it also has fatty acids, this whole thing is referred to as a phospholipid. The really cool thing about this phospholipid is that it means that the head is hydrophilic, so philia, water loving, and what that means is if you drop a bunch of phospholipids into water, what will eventually happen is that all of the nonpolar tails will aggregate together. So there's my nonpolar tails, and the polar heads will be oriented towards the outside. I'm just going to draw those as circles here. Okay, so the polar heads can interact with water on the outside, and inside we've got these non-polar tails. And these phospholipids can also self-assemble into a two-layer sheet. So here's my heads. There's my tails. And this two-layer sheet is referred to as a phospholipid bilayer. And we're going to be talking about phospholipid bilayers much more in the near future. Okay, so then there's one last category of lipids that I'm only going to briefly mention here, and those are the steroids. And I'm going to draw out an example of a steroid, cholesterol. And the main things I want you to notice here again is that this molecule mostly consists of C's and H's, and I've used a convention here that just implies that the H's are present. And so because it's composed mostly of C's and H's, it's nonpolar. Second, what's characteristic of this as a steroid is that it has this 4 sets of rings. And I want to say that although cholesterol tends to get a bad rap, it is incredibly important for cell function, so we will see it reappear briefly later on. And that's it for the lipids. So up next, we're going to move on to the proteins.